The following presentation is brought to you by Discovery Education, leading the world of digital and video learning. Discovery Education. Connect to a world of learning.
All of the countless living things that inhabit our world are made up of cells, the essential structural and functional units, frequently called the basic building blocks of life. Cells are often defined as the smallest units of life that can replicate themselves independently, because for the most part, they can't be seen without using a microscope. The microscopic view of the non-living onion cells seen here have been specially stained to highlight their features. As we can see, each cell consists of a small walled compartment, and it is because of this feature that they were given the name cells in the first place, over 350 years ago. Today, biologists classify cells into two basic types. The first are the simple prokaryotic cells, which lack a true nucleus, such as the cells of bacteria. The second are the much more complex eukaryotic cells, which have a true nucleus, such as this animal cell. The nucleus is a round, dense, membrane-enclosed structure that contains most of the cell's DNA, the deoxyribonucleic acid molecules containing the genes that regulate the cell's activities. The nucleus is surrounded by a substance known as cytoplasm, which is filled with an array of complicated structures needed to keep the cell functioning. The cytoplasm is always enclosed in a plasma membrane, also known as the cell membrane. In addition, certain types of cells, including those of plants, fungi, and bacteria, also possess a tough outer cell wall that surrounds the plasma membrane. The vast majority of prokaryotes, such as these bacteria, are microscopic, unicellular organisms, meaning they consist of just one cell without a nucleus. Some eukaryotic organisms, such as this tiny amoeba, are microscopic and unicellular too, but their cells possess a nucleus. However, most of the creatures that have nucleated cells, such as these trees, these fungi, these giraffes, and these fish, are made from millions, billions, or trillions of cells, and so are known as multicellular organisms. The diverse cells that make up the bodies of multicellular organisms usually develop from the single cell of a fertilized egg by a process known as cellular differentiation. The cells of multicelled organisms are often arranged into layers called tissues. The tissues are often arranged into complicated organs. And organs often work together with other organs to create organ systems to carry out difficult tasks such as digestion and blood circulation. It is important to note that viruses are in a category all their own. Much smaller than the smallest bacteria, viruses are not quite alive and aren't considered to be cells. Viruses possess genetic material and are capable of replicating themselves, but they can only do so by taking over the machinery of living host cells. So viruses represent a bridge between the living and non-living worlds. The nucleus is typically the largest single structure in eukaryotic cells and is often referred to as the control center of the cell, because it's where most of the cell's genetic instructions, its genes, are located. For the majority of its life cycle, nearly all of the cell's genetic material exists as a diffuse substance called chromatin, consisting of DNA, RNA, and protein that fills much of the interior of the nucleus. During cellular reproduction, when the cell starts to undergo mitosis, the chromatin condenses into well-defined chromosomes like these. The nucleus is separated from the cytoplasm by a double-layered nuclear membrane, or nuclear envelope, that is dotted with the tiny protein factories known as ribosomes. The nuclear membrane is continuous with a complex cytoplasmic structure known as the endoplasmic reticulum. Thousands of nuclear pores perforate the nuclear membrane and provide channels which allow for, and also control, the transport of molecules into and out of the nucleus. Many of these molecules consist of messenger RNA instructions from the DNA. The nucleolus is the round, dense structure shown here in the center of the nucleus. It produces the material from which the ribosomes are made. The nucleolus also plays a role in regulating cell division. The cytoplasm of eukaryotic cells consists largely of a gel-like matrix called cytosol, made up mostly of water and dissolved chemicals, that is frequently moving. The cytosol contains a cytoskeleton consisting of microfilaments, intermediate filaments, and microtubules, which are made from various proteins. These structures function to support the cell and also aid in movement. The cytosol is also packed with a variety of organelles. These are little membrane-bound cellular organs that carry out a wide variety of specialized tasks. In animal cells like this one, the organelles are the mitochondria, the centrosome, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi apparatus, peroxisomes, as well as numerous small vacuoles and lysosomes. Plant cells like this one possess all of the organelles of animal cells except lysosomes and centrosomes, but plant cells also have chloroplasts and a large central vacuole, which animal cells don't have. Mitochondria are organelles that play a critical role in generating chemical energy in the form of ATP from molecules of the sugar glucose and oxygen, a process known as aerobic cellular respiration. And it is for this reason that mitochondria are often referred to as the powerhouses of the cell. Cells can contain as few as one or as many as several thousand mitochondria, which move about inside the cytoplasm. Mitochondria contain DNA and self-replicate. This is one reason why many scientists believe that mitochondria were originally creatures similar to bacteria that were capable of carrying out the chemical reactions of cellular respiration. It is thought that many millions of years ago, one of these bacterial cells was engulfed by the ancestor of a modern-day eukaryote that at the time was unable to perform cellular respiration on its own. The bacterial cell remained in the host cytoplasm and reproduced. As a result, it gave rise to a permanent feature of eukaryotic cells, the mitochondria. The centrosome is an organelle found in animal cells that is composed of two centrioles. The centrosome's primary role is to produce the microtubules of the mitotic spindle that organize the chromosomes and pull them to opposite poles of the cell during cellular reproduction. The endoplasmic reticulum, or ER, is a network consisting of two types of flattened, membrane-enclosed passageways that are continuous with the nuclear envelope. The first type of ER, the rough endoplasmic reticulum, is covered with ribosomes that deposit newly manufactured proteins into its hollow center. The rough ER is a route taken primarily by protein molecules that are headed for specific destinations or require certain modifications. These proteins exit the rough ER inside of membrane-bound transport vesicles. The second type of endoplasmic reticulum, known as the smooth ER, lacks ribosomes and functions mainly in lipid and carbohydrate metabolism. In vertebrates, the smooth ER is a site of steroid hormone production. It also helps to detoxify drugs and poisons and stores calcium ions. The Golgi apparatus is an organelle found within the cytoplasm of both plant and animal cells that is composed of a series of membrane-bound sacs. Transport vesicles containing proteins made in the rough ER fuse with the Golgi apparatus. Inside the organelle, the proteins are modified, given molecular ID tags, and then shipped out into the cytoplasm in new vesicles, some of which are exported out of the cell so the products they contain can be used elsewhere in the organism. A good example of such export activity takes place in the human pancreas, where the Golgi apparatus in these cells plays a key role in processing, packaging, and transporting the hormone insulin, which is a very small protein or polypeptide. Insulin must be exported out of these cells so it can enter the bloodstream in order to reach the liver, where it regulates carbohydrate and fat metabolism. Peroxisomes and lysosomes are both small, roughly spherical, membrane-bound organelles. Peroxisomes get their name from the fact that they contain large amounts of hydrogen peroxide that is produced as a byproduct of breaking down fatty acids and toxic substances such as alcohol. Lysosomes, which are only found in animal cells, fuse with waste or food containing vacuoles. They release powerful digestive enzymes that cause the materials inside the vacuole to be broken down. A good example of this sort of activity takes place in infection-fighting white blood cells called macrophages that engulf disease-causing bacteria. The lysosomes fuse with the bacteria-containing vacuoles that are created. Their enzymes cause the bacteria to disintegrate, and eventually, the harmless materials are discharged from the cell. Chloroplasts are the green-colored organelles found in the cells of plants and algae, where the essential biological process of photosynthesis is carried out. During photosynthesis, chlorophyll molecules inside the chloroplasts trap light energy, which is utilized to combine carbon dioxide and water to create energy-rich carbohydrates, while releasing oxygen as a byproduct. The light-capturing chlorophyll molecules are located in a collection of disc-like sacs called thylakoids that are suspended in the fluid-filled stroma of the chloroplast. Chloroplasts move around inside of cells. In fact, under low-light conditions, they spread out to obtain the greatest amount of energy for photosynthesis. Chloroplasts possess their own DNA and can replicate themselves just like mitochondria, and they may have developed in a similar way to mitochondria too. Chloroplasts are believed to have arisen when an ancestral eukaryotic cell engulfed a photosynthetic bacterium, which remained in the cell and started reproducing. Vacuoles are the rounded, membrane-enclosed, liquid-filled compartments found in the cytoplasm of most living things. The green-colored food vacuoles in this amoeba, for example, are filled with plant material that it consumed by a process called phagocytosis, while the clear, contractile vacuole nearby pumps water out of the cell in order to maintain the proper osmotic balance of salts in the cytoplasm. In the cells of plants, the large central vacuole seen here typically makes up about 30% of the cell's volume. The central vacuole stores some of the water used in photosynthesis. It holds nutrients and wastes, and also acts to maintain the internal turgor pressure that's necessary for supporting the plant's leaves and stems. Ribosomes are the tiny molecular machines where proteins are synthesized in the cytoplasm. Ribosomes aren't actually considered to be organelles because they aren't enclosed in a membrane. They are found floating freely about in the cytoplasm of all cells, but they are also found attached to the rough endoplasmic reticulum and to the nuclear envelope in eukaryotic cells. Living things completely depend on the proteins that ribosomes make, because proteins form such things as muscle and connective tissues, as well as the many enzymes that are needed to keep organisms functioning properly. Proteins consist of chains of amino acid subunits that are arranged in a very definite order. For example, the hormone insulin diagrammed here is a very small protein made up of 2 chains of precisely ordered amino acids linked together. Insulin contains just 51 amino acids, but some proteins are made up of tens of thousands of them. The genetic code information for making proteins is stored in specific sequences of DNA called genes. Essentially, there is one gene for every protein or chain of a protein. The process of making proteins begins in the nucleus when the instructions for the protein are copied from the DNA into RNA by an event called transcription. The RNA is chopped up, parts are removed, and then it is spliced and reassembled into messenger RNA or mRNA. The mRNA is then exported through the nuclear pores and moves to the ribosomes, where the translation of its message into protein will occur. Ribosomes consist of 2 major components: a small ribosomal subunit, which reads the mRNA instructions, and a large ribosomal subunit, which joins amino acids together into a protein chain. Free amino acids are dissolved in the cytosol, but when a protein is being made, each one that's needed will be picked up by a specific transfer RNA, or tRNA. The tRNAs transfer the amino acids to the mRNA on the ribosome. Every time a tRNA anticodon matches with the proper mRNA codon, its amino acid will be connected to the amino acid that was added to the chain right before it. And this process continues until the complete chain of amino acids called for by the mRNA are linked together and a complete new protein molecule is formed. The plasma membrane is a thin, flexible structure that completely surrounds the cytoplasm. In animals and in protists, such as this amoeba, the plasma membrane is the outermost boundary of the cell and serves to separate and protect its contents from the external environment. Although a tough cell wall encases the cell membrane in bacteria as well as in plants and fungi. Additionally, in some cells, structures involved with movement, such as whip-like flagella and hair-like cilia, that are connected to the cytoskeleton, protrude through the cell wall and plasma membrane. The plasma membrane is a complicated structure consisting mainly of a double layer of phospholipid compounds along with cholesterol. Its outer surface contains a variety of carbohydrate side chains and receptor proteins that bind external molecules such as hormones. Embedded within the plasma membrane are a number of specialized channel protein molecules that form openings and create pumps that are used to move nutrients and cell products into and out of the cell. The plasma membrane is said to be semi-permeable due to the way that substances move across it. For example, it can carry out diffusion, in which small molecules like carbon dioxide and oxygen move passively across it in either direction. But the plasma membrane can also act as a barrier to certain ions and compounds. This results in the formation of an osmotic gradient of different concentrations of these substances on either side of the membrane. The plasma membrane can even physically wrap around and engulf materials and transport them into the cytoplasm that way. Cellular reproduction is crucial to the development and the maintenance of life on Earth. All multicellular organisms, from this sea turtle to this flowering plant and this monkey, began as just one cell. And after that single original cell divided over and over again, it ended up giving rise to an organism that consisted of a complex collection of billions or trillions of cells of many different specialized types, such as the nerve cells of the brain, the muscle cells of the heart, and the red blood cells of the circulatory system. In order for cells to reproduce, whether in a developing embryo or to replace worn-out cells in an adult organism, certain distinct steps must be followed to assure that the new cells will contain the same genes as the parent cell. In multicellular organisms, cell reproduction involves two basic processes. The first, which is called mitosis, is the duplication of the nucleus, the process of dividing up the cell's replicated DNA and distributing it equally into two new nuclei. The second part of cell division, called cytokinesis, starts near the end of mitosis. It is the process of dividing the cytoplasm in half to create two new and separate cells. Interphase is not considered to be a stage of either mitosis or cytokinesis, and yet it is the part of a cell's life cycle when it is preparing to reproduce. Interphase is by far the longest stage of the cycle, the period when the nucleus is intact and chromosomes are not yet visible. The cells in this stained slice of an onion root that possess round, intact nuclei are in interphase, while those where the chromosomes are visible are undergoing mitosis. The interphase portion of a cell's life cycle starts with the G1, or GAP1 phase, which is the 5 to 6 hour period following cytokinesis, when the cell grows to nearly its full size and starts to be busy carrying out its normal activities. The roughly 12-hour S, or synthesis phase, follows the G1 phase. This is the period of the cell cycle when the DNA is replicated. This process produces two complete sets of genetic instructions, one for each of the new daughter cells that will eventually be produced. The G2 or GAP2 phase is the final 4 to 6 hours of interphase, in which a second period of growth occurs. And G2 is when the newly duplicated organelles, called centrosomes, first appear in the cytoplasm of animal cells. Once G2 ends, interphase is over, and the cell begins the fairly short, roughly 80-minute M, or mitotic phase of its life cycle. Prophase is the first of the 5 phases of mitosis, which result in the duplication of the nucleus. At this stage in the cell's life cycle, the threads of DNA and protein, known as chromatin, found in the nuclei of interphase cells, starts to condense. The nucleolus disintegrates. The centrosomes migrate to opposite ends of the cell, sprouting tiny microtubules, and duplicated chromosomes arise from the chromatin. They are called duplicated chromosomes because their DNA was replicated during interphase. Closer inspection shows that they look different than normal chromosomes in that they are actually made up of 2 sister chromatids tightly joined together by cohesion and a structure called a centromere. The centromere that holds the sister chromatids together consists of 2 kinetocores, points where the microtubules will soon attach. As we can see, there are only 2 different types of duplicated chromosomes in this very simplified example: tall ones and short ones. But they are found in nearly identical homologous pairs. That's because the DNA information in one of the tall ones and one of the short ones is paternal and comes from the father, and the information stored in their opposites are maternal and come from the mother. When cells contain pairs of each type of chromosome, they are said to be diploid. Prometaphase is the second phase of mitosis. During the early part of prometaphase, the nuclear membrane disintegrates as elastic microtubules grow out from the centrosomes, forming a unique structure called the mitotic spindle. In typical cells, the complicated events of prophase and prometaphase take about 30 minutes to complete. Metaphase is the third and longest phase of mitosis, lasting roughly 20 minutes. This is the time in a cell's life cycle when the spindle fibers of microtubules have completely attached to the kinetochores of the chromosomes and has caused them to line up on the equator of the mitotic spindle, with the individual chromatids bending toward the opposite poles of the cell. Metaphase ends when the centromeres that bind the sister chromatids together break apart. Anaphase is the fourth and shortest phase of mitosis, usually lasting only 5 minutes. This is the time after the centromeres have split, when the sister chromatids are rapidly pulled apart by a shortening of the spindle fiber microtubules. At this point, the divided chromatids are true chromosomes called daughter chromosomes. Soon the spindle fibers disappear from view, and a full diploid set of chromosomes is now found at each end of the cell. Telophase is the fifth and final phase of mitosis. It is the time when the daughter chromosomes uncoil back into threads of chromatin. As new nuclear membranes begin to form around the chromatin, the cell elongates and nucleoli reappear, resulting in the production of 2 new genetically identical nuclei. And this completes the 5 phases of mitosis. In animal cells, cytokinesis, the division of the cytoplasm, begins during early anaphase with the formation of a ring of actin microfilaments called the contractile ring, which leads to the development of a cleavage furrow in the cell membrane during telophase. The compression eventually becomes so great that it causes the cell to divide in half. This completes the process of cytoplasmic division and cell reproduction, and the new cells return to interphase. Now let's see how some of the later events of cell reproduction look in a real living cell, one with numerous chromosomes. Here, we'll start at metaphase, with the duplicated chromosomes lined up in the center of the mitotic spindle. Then, as we enter anaphase, after the centromeres have split, the chromosomes are rapidly pulled toward opposite ends of the cell. Upon entering telophase, the chromosomes shrink and start to change back into chromatin. And as cytokinesis proceeds, the contractile ring slowly pinches the cytoplasm in two, creating 2 new daughter cells. The living cells of multicellular organisms vary tremendously as to how long they remain in the different phases of their life cycles. Nevertheless, all of them follow essentially the same steps when their cells are reproduced, even though the exact details differ somewhat among the kingdoms of living things. Interphase is the longest time period in the life cycle of cells, and the events of cellular reproduction completely depend on it. During interphase, the nucleus remains intact and distinct chromosomes are not yet present. Instead, the cell's DNA is found combined with proteins in loose threads called chromatin. Interphase is a time when cells grow, replicate their DNA, and perform their normal cell functions. In general, interphase takes up more than 90% of the cell's life cycle. When mitosis, the duplication of the nucleus, gets underway, the period of really active cell division begins. The first phase of mitosis is known as prophase. This is the time when the nucleolus vanishes and the duplicated chromosomes first begin to appear from the threads of chromatin. At this point, each duplicated chromosome consists of two sister chromatids attached together by a structure called a centromere. Prophase is followed by prometaphase, the second phase of mitosis, the time when the nuclear membrane disintegrates and the mitotic spindle forms from microtubules in the cytoplasm. In animal cells, but not plant and fungi cells, the spindle is organized by organelles called centrosomes. Prometaphase is followed by metaphase, the third and longest phase of mitosis. Metaphase finds the duplicated chromosomes lined up on the equator of the mitotic spindle. Anaphase, the fourth phase of mitosis, follows metaphase. Anaphase is the time in the cell cycle when the sister chromatids are pulled apart by the microtubule spindle fibers, and the new daughter chromosomes migrate toward opposite poles of the cell as the spindle fibers disappear from view. During telophase, the fifth phase of mitosis, the chromosomes unwind back into chromatin threads as new nucleoli appear and new nuclear membranes form around the chromatin. At this point, nuclear duplication is complete and mitosis has ended. Cytokinesis, the division of the cytoplasm, begins in early anaphase with the formation of a contractile ring. During telophase, the contractile ring squeezes down and creates a cleavage furrow, which causes the cytoplasm to divide, forming two new daughter cells that are genetically identical to the original parent cell, thus completing the process of cellular reproduction. Meiosis is a special type of cell division that is necessary for sexual reproduction to take place in eukaryotic organisms such as plants, animals, and fungi. During meiosis, the number of sets of chromosomes in the cell is reduced to half the original number, usually from 2 sets, or diploid, to 1 set, or haploid. The cells produced are known as gametes, or sex cells. Meiosis also provides a way for the DNA of the two parents to be recombined in new ways. And this helps to create genetic diversity in living things, which makes it possible for species to adapt to changes in their environments by allowing a few individuals to survive under challenging conditions. In creatures like these elephants, in which 2 parents are needed to create offspring via sexual reproduction, meiosis always begins in special diploid cells called germ cells. In female animals, the germ cells are found in reproductive organs called the ovaries, and in male animals, they are located in the testes. Immature germ cells, known as spermatogonia, divide to give rise to the mature male germ cells called primary spermatocytes, which can be easily seen in this microscopic image of a stained slice of tissue from the human testes. The mature germ cells will go through several stages of meiosis and cytokinesis to produce the haploid sperm cells visible in the center of the image. Before meiosis begins, however, the germ cells will spend a long time in an important state known as interphase. During interphase, the nuclei are round and intact, and the chromosomes are not yet visible. This is the time when the germ cell's DNA is replicated and is a period of active growth, when the cell manufactures a vast array of enzymes and structural proteins. During the first phase of meiosis, called prophase 1, a thickening and condensing of the long, twisted threads of a material called chromatin, consisting of DNA and protein, takes place in the nuclei of germ cells. And also during prophase 1 in animals, but not plants or fungi, organelles known as centrosomes migrate toward opposite poles of the cell, as the duplicated chromosomes emerge from the chromatin. Each duplicated chromosome consists of 2 sister chromatids that contain DNA replicated during the preceding interphase. The sister chromatids are joined together by cohesive forces and a structure called a centromere that is composed of 2 smaller structures called kinetochores. As prophase 1 progresses, the nucleolus and the nuclear membrane disappear, and a spindle of microtubules begins to form. As we can see, the nearly identical or homologous duplicated chromosomes have paired up with one another, so that, in this example, the tall chromosomes align gene to gene with their mates, and the short chromosomes do the same. This pairing of duplicated homologous chromosomes, known as synapsis, only occurs in meiosis and does not take place during mitosis. It is during synapsis that an important process called crossing over occurs, in which a random exchange of chromosomal material takes place between the maternal and paternal homologous chromosomes. This key event is one of the main things that produces genetic diversity among different offspring of the same parents. During metaphase 1, the second phase of meiosis, the homologous chromosomes line up on the equator of the cell with their centromeres aligned. In contrast, during mitosis, the centromeres align independently. During anaphase 1, the third phase of meiosis, the duplicated homologous chromosomes, each made up of sister chromatids, move apart, centromeres intact, toward opposite poles of the cell. But during mitosis, the centromeres pull apart to form daughter chromosomes. During telophase 1 and cytokinesis, the fourth phase of meiosis, each half of the cell contains a haploid set of duplicated chromosomes, each still consisting of 2 sister chromatids. Cytokinesis, the division of the cytoplasm, occurs simultaneously with the events of telophase 1, resulting in the formation of 2 daughter cells. Prophase 2, the fifth phase of meiosis, follows right on the heels of telophase 1, without an intervening interphase stage. As a result, the DNA is not replicated. Consequently, a haploid number of duplicated chromosomes is now present in each cell as a new spindle of microtubules forms. During metaphase 2, the sixth phase of meiosis, the microtubules of the spindle arrange the duplicated chromosomes so that they line up on the equator of the cell, and this time the centromeres do split apart. During anaphase 2, the seventh phase of meiosis, the microtubules pull the sister chromatids apart, and new daughter chromosomes are formed at each end of the cell. Finally, during telophase 2 and cytokinesis, the eighth and final phase of meiosis, the microtubules vanish, new nuclear membranes form, and nucleoli reappear in the 4 new nuclei, as cytokinesis cleaves the cells in two. As we can see, 4 new haploid sex cells have been produced from the single original diploid germ cell. So now, when 2 haploid sex cells join together, the fertilized egg will contain the same diploid number of chromosomes found in any body cell.